Okay, so today is all about gene expression and specialization. We talked about cell specialization when we talked about cell organelles back when we were in our cell biology unit. So today we're going to kind of talk about how our cells control that specialization through gene expression. So we're going to connect kind of a bunch of stuff all together at once. So, a lot of this is going to boil down to embryonic development because that's where a lot of this stuff takes place. It's where a lot of the differentiation happens, right? It's when lung cells become lung cells and heart cells become heart cells, and all of those cells get their specific jobs. And the process of going from a kind of blank slate embryonic stem cell to all of these other cells is called differentiation. That's the process by which cells become specialized. There it is. So I like this diagram right here. We don't have the opportunity to go too deep into embryonic development, but essentially, when an embryo is forming, we can divide cells into three layers: the mesoderm, the endoderm, and the ectoderm. And uh, making sure we have those three layers is super important. All of the mesoderm cells are going to be like cardiac muscle and skeletal muscle and red blood cells and smooth muscle. That's the mesoderm. The endoderm is going to be lungs, thyroid, hormone based organs, and the ectoderm is going to be skin cells, pigment cells, and neuron cells. So these three layers, and making sure these three layers exist early on in development, is super important. And that process of going from these stem cells to these kind of layered, uh, faded cells, and we'll talk about what faded means in a minute, is called differentiation. Aha. So, um, the process of going from, or the process of development, specifically with embryos and figuring out the structure of that embryo, is called morphogenesis. Um, and that morphogenesis is the development of the actual physical form and the structures of an embryo. So we've got like brain, spine, digestive system, those big structures, limbs, etc. And the way morphogenesis happens, or the kind of what morphogenesis relies on, is how these cells are organized. And there are a couple different physical things that can influence how these cells know which genes to express and then what type of cell to become. So, morphogenesis is the development of the physical form, um, and cell specialization is kind of how morphogenesis happens. So we've got this kind of seahorse looking thing, where we've got an area that becomes our skeleton for like brain support. Some of them become the skeleton for limb movement, and then one of them becomes the spine. And we've got all of these like special signals that get that embryo to be the shape that it needs to be. If you want a little evolutionary sidebar, the genes that control the general layout of an embryo can be a very interesting source of evidence for natural selection and evolution, because these genes across multiple organisms can be very similar, because the instructions for, okay, body, arm, leg, leg, right? A lot of mammals, especially, but a lot of organisms have that kind of body, four limbs layout. So it is believed that the genes that control that layout are from a common ancestor. So that's just kind of a connection in there. There are two different ways our cells know what type of genes to express. There are two big ways our cells in an embryo know, okay, I am going to be this type of cell. I'm going to be this type of cell. The first is the cytoplasmic determinants, and the second is its environment. So where in the embryo it is has a big effect. So we're going to talk about cytoplasmic determinants, and we're going to talk about how the environment can affect these as well. Questions so far? Doing okay? All right. So to start us off, we're going to talk about cytoplasmic determinants. They're essentially little molecules that are in the cytoplasm of the egg cell when it's fertilized. So there can be a lot of jokes to be made about the contribution an egg cell has and a sperm cell has to embryonic development, because the egg cell gives a lot more than just genetic information. We talked about how all of the initial cell organelles in a zygote come from the egg, and we also have these cytoplasmic determinants. When we've got an egg that is fertilized or not fertilized, it's got an uneven distribution of a bunch of different molecules. I.e., it is not homogeneous. There are mRNAs, proteins, and other substances that are distributed unevenly throughout the cell, and that is very, very important. These things, the RNAs, proteins, and other substances that influence embryonic development, are called cytoplasmic determinants. And I like this little clip arty thing here because it kind of shows a gradient. They fade into one another. And that, if you ever find yourself in a developmental biology class, you find out relatively quickly that most embryonic development is just based on concentration gradients, the amount of a certain substance in a certain location in a cell. So we've got a bunch of different proteins, a bunch of different RNA, and some other substances that are just kind of hanging out in their specific area of the egg cell. And those cytoplasmic determinants play a super, super big role in having an embryo develop properly. So not only does the egg give all of the cell organelles that zygote will need to survive, it also gives kind of a starting line for instructions on how to build that embryo from that first cell. Questions here? All right. So any cytoplasmic determinant can be like a number of things. It can be a protein, it can be a piece of mRNA, it can be some just like hormone chemical that's in a specific spot in this egg cell to give directions. The combination of these cytoplasmic determinants helps determine the fate of these cells in the embryo, by regulating the expression of genes. So right here, we've got our unfertilized egg, right? We've got these little triangle molecules, we've got these little spherical molecules, and there are way more than just two, but this diagram does this relatively simply. After that, that egg is fertilized, we've got a zygote, and then it will undergo its first mitotic division, split into two cells. And as you can see, right, we've got mostly those spherical cells over here and mostly those triangular cells over here, or molecules, sorry. And those molecules can regulate which genes are expressed in that cell and can kind of kick off hey, this cell might go on to be placenta, and this cell might go on to be actual embryo. There can also be gradients of molecules. We've mapped out almost with complete certainty the development of C. elegans, which is like a little wormy thing, and Drosophila, because we spend a lot of time with fruit flies. And we can actually trace the concentrations of different proteins throughout embryonic development and see what that protein does and which type of specialized cell it triggers. So it can even be like, oh, if it's got like 25 or like 25 molar this and 10 molar that, it's this cell. But if those concentrations are off just a little bit, it can be something completely different. It's very specific and kind of really cool. But there are other ways to be signaled. It's not just what's in the egg when it is fertilized. Cells can also receive signals, and we've seen diagrams like this before, right? Where we've got one cell signaling to another cell, like, hey, you need to start doing this instead. So if we've got cells forming a specific organ, and we need a different tissue in that organ, they can send that out and be like, hey, we've got this covered. You need to start being this. So it works just like the cell signaling that we talked about earlier this year. We've got our cells via exocytosis, right? Sending that signal out. The target cell has a receptor on it. That signal comes in. That cell's like, okay, here's my response, and starts triggering a new genetic change. These can send cells down specific developmental pathways. Meaning, once they receive these signals, they're on a path for good. And it is very, 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 very difficult, if not impossible, to get one of those cells to go back after it's reached a certain point. Questions here? All right. So, this process is called induction. It is very similar to signal transduction. We are inducing a cell to express a specific gene. Almost there. Give you one more second to finish up. All right. So, okay. Before we knew about a lot of the molecular biology that goes into development, all we knew, aw, is that one's gone for good? All we knew is that at some point during development, a point that biologists coined determination, all of the embryonic cells were irreversibly committed to becoming a specific cell type. There was a specific point at that we called determination, where after this kind of step in embryonic development, this cell was going to be a skin cell, this cell was going to be a heart cell, this cell was going to be a lung cell, and there is no going back. This point is called determination, and it's still a thing. We know a little bit more about it now because we know about the molecules that actually send these signals. But there's a really cool experiment they did to kind of figure out when specifically determination happened in different organisms. So this kind of showed them that there was a sequence to be had within embryonic development. This happened, and then this happened, and then this happened, and then this happened, and that process was always the same. And at one of those steps, everything had a fate. It knew where it was going to be and what it was going to be, and nothing could change its mind. So this, oh, come on. Apparently I didn't click it properly. Oh no. There we go. Oh, my clicker died. That's fun. All right. So, this right here is the normal sequence of events. We have these cells. They're all, we've got the green cells and the yellow cells. The yellow cells became these hexagon shapes, and the green cells became these square shapes. These are where we think these fates are determined, and this is after they have differentiated, right? What they did was they took a cell from this, or from the yellow is the word I couldn't think of just then, from the yellow section and put it in the green section, or vice versa. And if they did this before that determination point, those cells, those green cells that they brought up here, just followed everybody else's lead. Right? They became these little hexagons, even though they were originally in the squares. That's what happened if they took a cell out of the green part and put it in the yellow part before determination happened. They watched it and they said, oh, okay, everything is going just fine, everything is normal. Then they did the same thing after determination. They took a piece from the green section, brought it to the yellow section, and then watched it differentiate. And those green cells stayed the squares. They had gotten their instructions. They know what they're going to be. They're not going to disrupt the path. They became squares no matter what the outside world was telling them. So at some point in development, we reach this determination point, and this is where they figured out where that determination point was, because they just kept transferring tissues from different areas and watching what happened. Usually with Drosophila or C. elegans embryos, not with like mammal embryos. But this is how they figured out what determination was. Questions here? All right. So, fun. We can see, in combination with the cytoplasmic and induction signals, a plan forming relatively quickly. Both the inductive signals and the cytoplasmic signals contribute to spatial organization. That body here, leg here, head here, etc. Right? The general kind of body plan, or how these body plans came to be, is called pattern formation. Just like you were to make a pattern for a piece of clothing, right? You sew all the pieces together. The pattern is where stuff goes. This shows where stuff goes. So I've got a little Drosophila thing right here. We've got our fate map, right? So this part's going to become the proximal wing, the red part's going to become the distal wing, the blue part's going to become like the hinge of the wing, and over time, as that grows, we get this kind of format here. They did some really Frankensteinian things to fruit flies whenever they were trying to figure out determination. There's like two proteins that are in charge of determining where the butt of the fruit fly goes and where the head of the fruit fly goes. They realized that if they were to mess with the concentrations of these proteins, they could make a fruit fly with two heads and no butt, or just two butts. So I'll try to find those for you because they're actually kind of cool. All just by messing up with the proteins in the cells at a certain point. Yeah. We could technically try to make them. Let's see. Oh, I spelled that really wrongly. Where's it at? I'll find it in a minute. We have like one more slide. In addition to pattern formation, we've got the actual cues that help determine pattern formation, called positional information. So positional information gives our embryo the information it needs to create its pattern. This positional information tells a cell where it is relative to the body axis, right? So there are always signals being sent, and based on what signals a cell is receiving, they can be like, oh, I'm on the top half of this organism. Oh, I'm on the right side of this organism. It can tell where it is because cells don't have compasses and they don't need cardinal directions, but they do need to know where on the body it is. So there is information being sent constantly to figure out where that cell is, so it knows what to grow into. And as it figures out where it is, that's how we get pattern formation, where this cell is receiving this specific amount of signals. It knows it's on this side of the body at this point in time, so it knows it's going to become this section of a wing. So a lot of complex cell signaling in embryonic development. That involves a lot of genes we only use once. Once we've got the general body plan figured out, we don't need those genes anymore. But we do need to keep them around so that if an organism ever procreates, it can like send that information to the embryo. So positional information helps a cell figure out where it is in an embryo, and it can create patterns through that. And I've got a picture for you. Right. So this right here is a fate map of a seedling, right? So as a seedling is growing out of a seed, we've got that zygote, that seed, right? And it's got a bunch of different signals coming from the embryo sac. It knows it's got these signals over here, signals in the middle, signals up top. The zygote is inside of that. Over time, these signals, as that cell, that single cell divides, the cells towards the top are getting these signals, the cells towards the bottom are getting these signals, and we can see over time how these signals change and how it can lead from one cell to 16 cells to being an actual seedling. This part's going to become the root tip, this part's going to become the root, this part's going to become the stem, this kind of leaves out, and these are those first little leaves. So even from being a singular cell, it knows how, as it grows, to become its initial form. Which is kind of cool. Questions here? All right, that's what I got for you. That is cell specialization and cell signaling.